Welcome to another video in the series about synthetic biology. In the last video you learned about gel electrophoresis. Now we will discuss other methods and tools used in synthetic biology, specifically polymerase chain reaction or PCR and the centrifuge. The goal of PCR is to replicate or amplify template DNA strands so they can be extensively studied and used, such as being cloned in a plasmid or sent for sequencing. The components used in PCR are RNA primers, which attach to the beginning of the template strand, and DNA polymerase, specifically Taq polymerase, a heat-resistant enzyme that adds complementary free nucleotides to the template strand, creating a complementary strand. The first step of PCR is denaturation, in which a double strand of DNA is separated by raising the temperature of the mixture until the thermal energy breaks the hydrogen bonds between the nucleotides which hold the two strands together. This usually occurs around 94 to 98 degrees Celsius. At this point, the two strands can be separated. The mixture is then cooled down to around 50 to 65 degrees Celsius, and the second step begins: annealing. One primer attaches onto the 3' end of each template strand, pairing complementary nucleotides to the template strand to begin the 5' end of the new strand. These primers are necessary to begin step 3, extension. First, the mixture is heated to around 68 to 72 degrees Celsius. The DNA polymerase then attaches onto each template strand and extends the new complementary strand by adding nucleotides that form hydrogen bonds with those on the template strand, adenine with thymine and cytosine with guanine. The DNA polymerase continues adding nucleotides until it reaches the 3' end of each complementary strand. At that point, one cycle of PCR is complete, creating two double strands of DNA which are identical to the original. A PCR reaction usually completes around 25 to 35 cycles, and because each cycle doubles the amount of DNA, the strands exponentially grow with each consecutive cycle. Next, the centrifuge is a rotating device that separates molecules by their densities. First, you insert a liquid mixture, such as blood, into a test tube, which is then inserted into the centrifuge. As the centrifuge rotates in a circular path, the molecules in the mixture will constantly be changing direction. Denser molecules will be more resistant to this change, or have a greater inertia, while smaller, lighter molecules will have less inertia. As the densest molecules resist a change in the direction of their movement, they move to the outside of the test tube, pushing less dense molecules to the inside. After the process finishes, the molecules in the test tube will be separated, with the least dense molecules on one end and the most dense on the other. This process is extremely useful as it can be used to detect certain viruses, bacteria, or parasites in a liquid medium, based on their unique density compared to other human cells. The same goes for DNA or protein, which can be detected in a mixture of other macromolecules. That's it for this video on PCR and the centrifuge. Make sure to come back for the next topic, RNA interference.